All right, it is 2, so we will get started here. Hopefully you watched the videos I put up on Friday. Again, apologize for canceling at the last minute there. But on Friday, we continued our discussion on sugars. So we talked about the monosaccharides, and then how we can combine monosaccharides into disaccharides. And there we talked about glycosidic bonds, we talked about the linkages between sugars, and we talked about how to name our glycosidic bonds. If you have questions about that, please don't hesitate to reach out, and I'll be happy to either meet one-on-one or via email exchange about those topics. But with that, we are going to continue on today with our discussion on sugars, and we're going to talk about sugar and proteins coming together. And that's what we call glycoproteins. So if we look at the word glycoproteins, glyco, anytime you see that, it means sugar, so sugar proteins. And for eukaryotes, most of our proteins are glycoproteins. That's one of the big differences between prokaryotes and eukaryotes. Prokaryotes, your bacteria, they really don't have glycosylated proteins, or glycoproteins rather, while eukaryotes do. And the weight due to these sugars can vary drastically depending on what protein you're looking at. As I have here, the sugars can take up anywhere from 1 to 90% of the weight of the protein. So just maybe 1 or 2 sugar units to a lot of different sugar units. And the really weird thing about these glycoproteins is this idea of microheterogeneity. What that means is, let's say you have 100 copies of the same protein and they all get sugars on them. Microheterogeneity means that the amount of sugars and the type of sugars these proteins get will be different, right? They're not going to be all the same. They're going to be slightly different here and there. And which is really weird because the amino acids themselves in the protein will be 100% identical. The structure of these proteins will be completely identical as well. But there is a difference when it comes to what sugars are on these proteins. And this field of studying glycoproteins and sugars itself is called glycomics, the study of carbohydrates. Now we're going to focus in on one type of glycoprotein to begin with, and then it's a massive one, and they're called Proteoglycan. Now, proteoglycans are like a class of proteins. They're proteins plus these glucosaminoglycans. And those glucosaminoglycans, we talked about that on Friday. They're those disaccharide units with amines and sulfurs on them, very highly negatively charged, and it's just repeats of disaccharides. And like I said in Friday's video, these are usually found outside the cell, and that's what this is, in the extracellular matrix. And these are made up of basically 4 things. You have your protein component. That's the black lines here. So all of these black lines are proteins, and it's called the core protein. Off of these proteins, you have different glucosaminoglycans. Well, in this case, you have 2 different types. The green are keratan sulfates, and the blue are chondroitin sulfates. So very highly negatively charged sugars on these proteins. This big red thing is actually sugar. This is actually a carbohydrate. It's not a protein. It's hyaluronate. This is one of the disaccharides that was mentioned on Friday. And then connecting your core protein to your hyaluronate is what we call link protein, not proteinin. I should say protein. So I have a picture on the next slide that looks over this in more detail so we can get a better image. But these things are massive. This is actually a microscopic version of it. And it's hard to tell size if you're not used to looking at pictures like these, but this is a very, very, very big complex, like bigger than cells, much bigger than cells. So let's take a zoomed in look on our proteoglycan. So this image in your bottom left, that's the same image I was showing on the previous slide. Right? So the red part again is our hyaluronate. And again, it's just a disaccharide that repeats up to 50,000 times. So a lot of sugars. The thinner red lines, that's your core protein. This yellow is our linker protein, which helps to connect our core protein to our hyaluronate. Blue here is our keratin sugars, and our red are our chondroitin sugars. So how are we connecting our proteins and our sugars together in glycoproteins, and specifically our proteoglycans? Well, the glycosidic bond. So the glycosidic bond is what we talked about when we were discussing how sugars are connected to each other. We call a sugar protein bond the same thing, a glycosidic bond. And you can have two types of glycosidic bonds, and you have to pick like either one. You're either an N-linked glycosidic bond or an O-linked glycosidic bond. Now for disaccharides, like was on Friday again, we looked at the O-linked disaccharide bonds. They're usually always connected via O-linked, and O-linked just means that it has an oxygen in the bond. And proteins can be connected to carbohydrates using an O-linked glycosidic bond as well. And they use side chains of amino acids. So either serine or threonine, as shown here, can be used to be connected to sugars via this glycosidic bond. Or you can have an N-link glycosidic bond, as shown in the picture below. This is also how nucleotides, how those are connected to their sugar bases, right? Your A, T, C, and G are connected to sugars via N-link glycosidic bonds. For proteins, you're going to be using asparagine here because it has that nitrogen at the end. Not arginine or it's just asparagine for that. Now, if we look again at our example of our proteoglycan here, I said up to 50,000. Well, it varies. That's why it's up to. And so the length can be anywhere from 4,000 to 40,000 angstroms, which is 400 to 4,000 nanometers. So sounds small, but on a cellular scale, that's very big. And depending on the size, you can have a different, like up to 100 different core proteins. And to really think about what this is doing, they're almost like pipe cleaners, and they actually look like pipe cleaners. So think about the core protein as like little pipe cleaners that are coming off the center carbohydrate, right? And each one of these core proteins, each one of these pipe cleaners can have roughly 50 keratins and 100 chondroitin. And if you remember from Friday, all of these are very negatively charged. So you just have a huge thing of negative charge, which is going to attract water. And if we actually look at it in your extracellular matrix right here, the green here is our hyaluronate. The black lines are core protein with our different sugars. It's going to interact with collagen. And we talked about collagen already a few weeks ago. That's a protein that forms a helix. And all of this is going to be very hydrated. And that is your cartilage, right? Your cartilage is basically made out of these simple components. It forms that gel because all of that water and sugar. And I think I mentioned this on Friday as well, but the reason why these are so good at providing cushions is because they are hydrated. And then when you try to compress it, since all the sugars are negatively charged, they will try to repel from each other to try and keep that cushion intact. So that's why you can jump up and down and not blow out your knees immediately, because these sugars are repelling each other. So there's the structure and the function of our proteoglycans. And what I really want you to really take away from this slide is how the structure can really dictate the function. Like, how is the structure of proteoglycans that we talked about here really related to their function as being part of the cartilage? And that's what I mainly focus on in my discussion there. So before I move on, is there any questions at all about anything I went over as I go and close my window so you don't continuously hear birds? Right? No questions? We can move on then. Oops. Yeah. So let's check our understanding then. So following the synthesis of proteoglycans, many of them are stored in intercellular compartments, and they contain a lot of calcium ions, which are rapidly pumped out as the proteoglycans are released extracellular. What is the function of these calciums? So anyone have any ideas about that? And I'll just draw a picture, right? So this is a cell. Let's say your proteoglycans are stored here with a bunch of calciums. Then when the proteoglycan starts to expand in the EC matrix, the calciums are removed. But they go away. Bye-bye calciums. What is the function of those calcium ions then? Why are they there to begin with, and then why are they removed based on what we just talked about in the last slide? They help maintain, do they help maintain the condensed state of the molecule? Yeah, they do. Because when you're creating it, you don't want it to be expanded. And so if these sugars have these extremely negative charges, which they do, you want it, and if you want this sugar to be condensed, you have to balance out these negative charges. So an easy way to do that is just make sure there's a lot of positive ions near it, like calcium. This calcium will be attracted by the negative charges, allowing this thing to remain small and condensed. And then when you pump it into the extracellular matrix, you want it to expand. So you want the negative charges to repel each other. Well, to do that, you have to remove the calcium ion. So when you're ready for expansion, you'll start to just pump out the calcium ions and release this proteoglycan into the extracellular matrix. All the negative charges now feel each other, they expand, and you make your gel. So that's what's going on there with calcium. And then similar to A, just understanding like the structure of these molecules, on the extracellular matrix, we have different things that will interact with our proteoglycans. One of them being fibroblast growth factors, which will bind to our glycoaminoglycans. Therefore, what amino acids are probably there? Well, like I've been alluding to a lot, these are very negative, so you want your positive amino acids. So these fibroblast growth factors, you would expect them to have R, K, and H molecules, right? Arginine, lysines, and histidines to be able to bind to these negative proteoglycans. All right, so any questions so far about anything we have covered? All right. So question 2, let's say you're doing SCS page. And when you do SCS page on a glycoprotein, you are more likely to come out as a smear. What I mean by that is you take a sample, let's say you purified like 1,000 proteins or 1,000 of the same proteins, but they're a glycoprotein. And normally you would expect a nice sharp band showing what the way the protein is. But glycoproteins can smear like this, where you just tape up a bigger weight. Why are glycoproteins more likely to be a smear and non-glycoproteins are more likely to be a nice sharp band? What can explain that we talked about on one of the first slides for today? If you're not quite sure, go back to the first slide and see if you can connect that idea. And just remember, SDS page is measuring molecular weight. So why do glycoproteins look like they have more varied molecular weight than a non-glycoprotein? Anyone have any ideas? And if you're not sure, just go back to our first slide and try to marry this idea to what we talked about there. And I'll give you a hint. The word I'm looking for starts with an N. Ends with a Y. There's a hetero in here. Yeah, there we go. Microheterogeneity. All right, so let's go over this concept one more time. Let's imagine that you have, and I'll make it simple. You have 5 copies, 5 identical copies of a protein. I'm going to call it protein A. And protein A is a non-glycoprotein. Therefore, if I measure the weight of protein A, let's say the first copy weighs 45 kilodaltons, 45.0 kilodaltons. That's A1, copy A1. Copy A2, I would then expect to weigh 45 kilodaltons. Copy A3 would weigh 45 kilodaltons. The fourth copy of that protein would weigh 45 kilodaltons. The fifth copy of that protein would weigh, you guessed it, 45 kilodaltons. So if a protein doesn't have any sugars on it, it's going to weigh the same exact thing, no matter what copy you look at, because it's made out of the same amino acids. It does not change. However, protein B is a glycoprotein. So let's say copy B1 weighs, I don't know, 31 kilodaltons. Well, the second copy could weigh 31.1 kilodaltons. The third copy could weigh 29.9 kilodaltons. The fourth copy could weigh 31.4 kilodaltons. Fifth copy could weigh 29.9 kilodaltons. And the reason why a glycoprotein will vary in the weight is microheterogeneity. When you put sugars on a protein, you don't put the same exact sugars on the same protein or the copies of that protein. They can vary slightly, which is why when you look at their weights, if you're looking at 1,000 copies of a glycoprotein, their weights can all be slightly different, which would explain the smear. While on a non-glycoproteins, the weights are going to be the same. They're always going to be the same, so you get a nice crisp band there. So any questions about that idea? All right, moving on then. Now let's talk about glycoproteins in context of bacteria. And I did say at the very beginning that bacteria don't add sugars to their proteins, don't glycosylate their proteins, but they do have a sugar protein matrix. And we call that the Peptico Glycan wall. Again, look at this word anytime you see peptide, protein, glycan, sugar. This is your protein sugar wall. And this, depending on what type of bacteria you're looking at, if you're looking at a gram-positive bacteria, it's going to be your outermost wall. If you're looking at gram-negative bacteria, it's going to be between your outer membrane and your inner membrane, or your plasma membrane and your outer membrane. So gram-positive bacteria have a much thicker Peptico Glycan than the gram-negative. But the gram-negatives, those are the ones that our bacteria are harder to kill, because this outer membrane helps to protect them more. So gram-negatives are the ones that are usually more resistant to antibiotics, and those are the ones that really give researchers trouble in trying to kill them, because this outer membrane can help protect the cell from antibiotics. Now let's take a deeper look at what this Peptico Glycan is, though. What is surrounding these cells? And I really think of the Peptico Glycan as like a sugar protein net with holes in it. And the holes are random. But before we get into that, let's actually look at what makes up the Peptico Glycan layer. So the two sugars that repeat, the disaccharide unit of the Peptico Glycan, are N-acetylglucosamine. So let's break this down. Gluco, that means it's glucose. N-acetyl means you have a nitrogen in an acetyl group right there. And the amine is NH2 or NH in this case. So N-acetylglucosamine and N-acetylmuramic acid. So this is just a different sugar than glucose, one we really haven't looked at. But those are your two repeating units here. And that's being shown down here. Here's just a mock-up of what the Peptico Glycan kind of looks like. Orange and yellow represent our two sugars. On your muramic acid, you have amino acids that are linked in an O-linked glycosidic bond. Here, it almost looks like threonine that we're connected to our glycosidic bond here. And off of this glycosidic bond, you have amino acids. You have alanine, isoglutamal, something we've never talked about. What isoglutamal is, it's a non-standard amino acid that's almost like glutamic acid, but not quite, because the difference is that you have an isopeptide bond. And an isopeptide bond is a peptide bond that uses a side chain instead of a backbone. So just as a reminder, as it's been a while since we talked about protein structure, so there's glutamic acid. This is glue. All right, backbone, side chain. Here, we have lost one of the oxygens to make a glycosidic bond. And so we are connected to lysine. And then something weird, we have D-alanine. Now, 99.9% of the time, amino acids are in the L conformation. In the Peptico Glycan, though, we have amino acids in the D conformation. And in this picture, we always have 4 amino acids. In reality, this can vary anywhere between 3 and 5, and they come in different like percentages. But to simplify things, we're just looking at 4 amino acids here. And this is the way to look at this. Here's an image: is that you have sugars that are kind of running parallel to each other, and they are cross-linked roughly 40% of the time. So let me get a different pen, different color. So this red is supposed to be representing like cross-links between the strands. And so not every connection point is cross-linked or connected to each other, like shown in this picture. They're more random than that, more dispersed. So you get this net that's full of big holes. And this is kind of allowing things to come in and out of the cell. Now, for a bacteria to live, you need a Peptico Glycan layer surrounding it. And one way to kill a bacteria is to destroy the Peptico Glycan layer. And that's what the protein lysozyme does. Lysozyme is found in your tears. And what it does is that it breaks the beta-1,4 glycosidic linkage between N-acetylmuronic acid and N-acetylglucosamine. So we're breaking that beta-1,4 bond. So we're just chopping apart all of these bonds, which causes the Peptico Glycan to fall apart. However, if you're a gram-negative bacteria, which I'll just go back to. If you're a gram-negative bacteria, you have this outer membrane that protects your Peptico Glycan layer. So we can't really cut it up with lysozyme. So those bacteria stay around. Early in the 20th century, we found another material that will kill the Peptico Glycan layer called penicillin. And what penicillin does is that it will go and bind to the enzyme that does the cross-linking. And so penicillin stops these cross-links from being made. Again, the Peptico Glycan layer doesn't form correctly, and the bacteria dies. But if you're gram-negative, you aren't affected by penicillin. And in fact, a lot of bacteria now, because of the widespread use and misuse of penicillin, now have their own enzymes that will go and deactivate penicillin. They'll take the penicillin molecule and just destroy it before it ever affects them. So that is just kind of like the war we have with bacteria. We develop something, they find some way to counter that. And that's also one of the reasons why like D-alanine might be part of the Peptico Glycan layer, because you can imagine, thousands, millions of years ago, there was an enzyme that might have like destroyed the linkage between lysine and alanine here and destroyed the Peptico Glycan layer, or just destroyed somewhere on this tetrapeptide. And then some bacteria created D-alanine instead of L-alanine, and enzymes generally can't work on D amino acids, so whatever enzyme was working on those amino acids no longer work. So that's just one way or one explanation why D-alanine might be part of the Peptico Glycan layer as a defense mechanism. Right, so any ideas about the Peptico Glycan layer or anything else up to this point? Right. So here's a little self-test for you, just to make sure that you got it, and also a good thing to come back to. So which one is not a structural component of Peptico Glycan? B, what feature of the bacteria Peptico Glycan is not essential for structural stability? So take a minute or two, look back in the previous slide and see if you can answer this, and I'll be back in a minute to give you the answers. But yeah, little review time. So do have a question about gram-negative bacteria. Can the membrane that protects gram-negative bacteria be broken down to better reach them? So this is not something I've really looked into all that much. My guess would be yes, because it's just membranes, but I'm guessing that it's hard to do. So since I don't really know much about that, this is a good extra credit opportunity. So if people, and this is open to anybody, if you want to send me just like a half a page or whatever with references on how to attack gram-negative bacteria, I think that would be a worthwhile thing to look up, and let me know. And I'll be happy to give you some extra credit on that, since I do not. My guess would be yes, but I don't know how to really do that, as I'm not that well versed in antibiotics. I just know the basics of them. All right, so let's look at this question then. So which of these is not a structural component of Peptico Glycan? Well, they are made out of disaccharide units, so it's not B. These disaccharyl units have N-acetyls on them, so it's not C. And connected to these disaccharides are tetrapeptides, so it's not D. So it's A. So in your peptoglycans, there's not like sulfates on your sugars. These aren't glycoproteins like we talked in the eukaryotes. B, what feature of the bacteria peptico glycan is not essential? N-acetylmurmonic acid. Well, that is one of the disaccharides, so it's not that. D amino acids. Well, this is one of the few times where we do have D amino acids. And we also have L amino acids. So for B, the answer here would be D. We don't have really monosaccharides part of our peptico glycan. They are disaccharides. So this was just a little review on the peptico glycan layer. So any questions about that? All right. And yeah, so this is our last question. Then we go. So what is the point of peptico glycan layer having holes? Because remember, it's only cross-linked like 40% of the time. So I'm going to redraw my picture. It's like a giant, it's like a net with just random holes in it. What's the point of those holes? And remember where peptico glycan is situated. It's outside the cell. So why would we have giant holes outside the cell? And one way to think about this, I suppose, is let's say we have two cells. So the black part's your cell. The red part's going to be Peptico Glycan. I'm just going to draw it as a straight line. So this one has a Peptico Glycan with holes in it. It would really be like a mesh work, but that's easier to do. This one doesn't have holes in it. What's the advantage of having holes? Yeah, so things can reach the cell through the pores, exactly. So a cell is isolated and it needs to exchange materials with the outside world. So if you had like an outer meshwork that didn't let anything through, you couldn't get materials and you couldn't expel toxins. So that would be very bad news for that bacteria. So you do need holes to let things in and out. So that's why the Peptico Glycan is porous to begin with. It's there for structural components and also has regulation purposes, but you need holes in it to let stuff through. And B, what is the advantage of having D amino acids? Again, it's for antibiotic resistance. 99.9% of our amino acids are L, but if you have a D amino acid, enzymes can't work on D amino acids, so those enzymes are stuck. And during that, I did just do a little Google search. And breaking the outer membrane of cell gram-negative bacteria, it's very hard to do. And it's just a constant area of research. And there are ways to do it, but bacteria become resistant to that. But my offer will still be open. If you want to do a little digging on yourself, educate yourself on that and just send me what you find with some research references. And the references need to be either from like a .gov website or scientific papers. I don't want any .coms because you can't trust .coms. Usually it has to be usually a .gov or .edu would suffice for that. Tell me about what techniques people are looking for to break down the outer membrane of gram-negative. I can give you a couple extra credit points for that. But with that, that's all we had to cover for today. So any questions from anybody? Right? If not, I'll put a homework up like always. Just as a reminder, if you haven't heard in the news, anyone over 16 can now get their COVID shot. So I would strongly recommend you try to do that ASAP, especially since we're looking to come back in the fall, if you're still going to be attending school in the fall, that is. Otherwise, I hope you have a good rest of your Monday. If you have questions about anything, never hesitate to reach out. And hopefully I'll see you on Wednesday. Have a good one, everybody.